I'm going to just spend maybe 10 minutes talking about mucoepidermoid carcinoma, and this is a presentation very much from a a clinician's perspective, just talking about some of the clinical considerations around the disease and how we manage it, and particularly from my perspective as a medical oncologist. And I'll explain a bit more about that in a second. So just to outline, we're talking about mucoepidermoid carcinoma, which is a subset of salivary cancer. So here we have just a figure showing different bits of anatomy of the head and neck, and you can see we have these three major salivary glands, or three pairs of major salivary glands: the parotid, submandibular, and sublingual glands. So most patients with mucoepidermoid carcinoma, it arises in one of these three pairs of major salivary glands, but you can also get mucoepidermoid carcinoma in other smaller salivary structures of the head and neck, and in fact in other exocrine secretory structures throughout the body, so other bits of the body, not just the head and neck. And so I often show this slide at the start of a talk, just to highlight one of the sort of challenges, and also maybe present some of the context of different types of salivary cancer. So there are many different subtypes of salivary cancer. We often say 24, but there's probably many more than 24. You can keep sort of subclassifying and subclassifying. And mucoepidermoid carcinoma is shown here, representing around a fifth of all new cases of salivary cancers. And this is from the UK Cancer Registry data that was actually published a few years ago now. And it shows that when they collected this, this was 4 years worth of data through to 2013, and it was only 3,500 cases in our cancer registry of salivary cancers. And so it highlights some of the challenges we have in both the management of salivary cancer and also in research into salivary cancers. The small absolute numbers make it a very difficult disease to standardize management across multiple hospitals, and also make it a difficult disease to recruit enough patients to get big enough groups of patients to actually develop research around. So I'm just going to present, this is from our medical oncology practice. So one of the questions we have for our patients with metastatic salivary cancers is how do we manage them when it comes to different drug therapies? So the reason that is my interest, just to explain, is as a medical oncologist, my focus is around drug therapy management and research. So I'm part of a team. We have surgical oncologists or surgeons that will resect a localized tumor. We have radiation oncologists, although in the UK called clinical oncologists, who will irradiate either a tumor or give that postoperatively, and then medical oncologists that focus on drug therapy management and research. And so that's my focus for the next few slides, is what do we know about and how do we manage these patients with drug therapies. And so one of our approaches is we look at the tumor DNA to identify different mutations that might help identify trial therapies. The reason we do that is there are no standard drug therapies for patients with mucoepidermoid carcinoma. So you can see here, this is our first 209 patients that we included. This was a number of years ago we published this, and we've got all of our subtypes shown at the bottom here. And actually the MEC is our mucoepidermoid carcinoma abbreviation. So of 209 patients that we sequenced the DNA in the tumor to look for different changes, we only had 7 with mucoepidermoid carcinoma. Now, for patients with mucoepidermoid carcinoma, that's actually quite a reassuring thing. And the reason is the patients I see have predominantly recurrent or metastatic disease. So 190 of 209 patients here had recurrent disease. The reason then only 7 of them, so 3% of my patients have mucoepidermoid carcinoma, is because disproportionately they don't get recurrent or metastatic disease compared to other subtypes, such as here ACC or adenoid cystic carcinoma, where we have 142 of our patients fell into that category. So I think that should be a reassuring thing, in that if you've had a resection, the chance of recurrence is much lower compared to other subtypes. However, it does pose an extra challenge, which is there are fewer patients then to recruit into clinical trials, and that's why we're still sort of behind other more common tumour types with our knowledge of what are the best treatments. And so this is just to demonstrate these are the different genes shown here in our panel that we'd included, and then here the third column just showing these trials, or with their abbreviation, the NCT clinical trial abbreviation, showing that most of the genes we included in the panel had clinical trials that they would match patients to. And then you can see here, so our 6 patients, that were included with mucoepidermoid carcinoma that had a valid result. So you can, the way this is shown, you can see each column represents an individual patient. So this box here in the lower panel B, MEC, mucoepidermoid carcinoma, so each column with a grey dashed line running top to bottom shows the different, so it's an individual patient, and you can see 6 columns. And actually in that panel, all that was identified were two mutations in TP53. And on the previous slide, you can see at the bottom is TP53. That was one of the mutations we actually didn't have a clinical trial for. So using this approach for our relatively small number of patients, we didn't actually manage to identify any matched clinical trials. I'll skip past this slide. But then the next approach we've got is looking for whole genome sequencing. So if you look for mutations in one of these 24 genes and don't find it, it doesn't mean that there aren't potentially druggable changes in the DNA of those patients. But what it does mean is we don't know what those might be. And so another approach is to look at every gene in the genome. Now, we can't, so for a period of time, around 12 months ago, we were doing that routinely for our patients with recurrent or metastatic salivary cancers. And we included a cohort, our first 14 in this analysis here. Now you can see none of these patients were initially classified as mucoepidermoid carcinoma. You might expect that because it was a relatively, it was only, I think, 3% of our patients had mucoepidermoid carcinoma. But it turned out when we did our analysis, I'll skip on to the next slide. One of our patients we found here did have a gene fusion in MAML2, MAML2 gene fusion. So that's this patient here, patient 13, that had previously been classified as having an myoepithelial carcinoma. And in fact, the presence of a MAML2 fusion meant that that patient was reclassified as having a mucoepidermoid carcinoma. Now that will lead quite nicely, I hope, onto the next the next presentation, where Lynn Bingle will be talking a bit more about MAML2 fusions. And then this is just to demonstrate from a histological perspective, our pathologist who's looked down the microscope, this is what the sort of images they look at when they stain the tumor samples down the microscope. And I think this at the top shows what was a clear cell carcinoma that was then reclassified from a epithelial myoepithelial to a high grade clear cell mucoepidermoid carcinoma based on this MAML2 fusion. And I do have one other slide. So it wasn't relevant to our patient with a mucoepidermoid carcinoma, but we also found this other mutation or gene fusion in FGFR1. So one of our 14 patients. And the exciting thing about that for that one patient was that there is an FGFR inhibitor, so a targeted therapy that we treated this patient with. And you can see this scan at baseline showing lots of tumor here. So this is fluid and tumor around the right lung and fluid here. And then at month 8, you can see complete resolution. And he had about almost 2 years of really meaningful benefit and response to this FGFR inhibitor, which we were able to treat him with, having identified this new fusion on his whole genome sequencing. So I was going to end my presentation there, but I think particularly the MAML2 fusion hopefully leads into our next presentation this afternoon.
So I am a non-clinical bod, a non-clinical scientist who has an interest in salivary gland cancers. So we've been doing some work looking at mucoepidermoid cancers from a perspective of, I work in a dental school, and so we're obviously very interested in head and neck cancers, but I have a particular interest in salivary gland cancers. And I think because there's, as Rob has suggested, they're quite rare cancers, so it's actually quite difficult. And so the first little presentation that I have for you, the first study that we did, was actually diagnosing the cancer can be quite difficult. And even though I work with specialist oral pathologists, I always jokingly say that if we give them 5 cancers to diagnose, and there's 4 pathologists in the room, we'll get 6 diagnoses at the minimum. And it's because, as Rob showed in one of his slides, you know, the histological structure of the tumors is very, very different and difficult. And there's not one classic mucoepidermoid structure, and that makes it quite hard. And as I said, I'm working with specialist oral pathologists who look at these things relatively regularly, with the caveat that they're rare cancers, but they are looking at them relatively regularly. But if you happen to be being treated initially in a non-specialist center, and the general pathologist is trying to make a diagnosis, it can be extremely difficult for them. And so using the MEC, or it used to be called MEC mammal, and I keep calling it MEC mammal. It's not, it's a CRTC1 mammal 2 fusion gene that's associated with mucoepidermoid carcinomas. And in many ways, I've looked at mucoepidermoids and I've looked at adenoid cystic carcinomas, and the fusion gene associated with adenoid cystic carcinomas is much more complex. The genetic alterations are much more complex. So in retrospect, starting with the mucoepidermoids was a good starting point. So it's always the same genetic rearrangement with a CRTC1 mammal 2, which does make things slightly easier for someone like me, a molecular and cell biologist, who's actually trying to locate that fusion gene and see if we can use that as potentially a diagnostic tool to help those non-specialist pathologists and even the specialist pathologists, but also could it be used as a prognostic tool? And it is interesting because obviously, well, maybe not obviously, but a fusion gene is a combination of 2 genes. So part of one gene coming together with part of another gene, and that quite often has downstream effects. And often it causes or has the potential to cause inappropriate cell growth, resulting in your tumor, because of the genes that are involved and the downstream effects that they normally have. We don't know that for sure with the mucoepidermoids, but we assume that that's what the fusion gene is producing. Could we use it as a diagnostic or a prognostic tool? Wonderful, thank you. So this is just a schematic of 2 genes, the CRTC1 and the mammal 2. And they come to, you know, I've put them in red and green. The CRTC1 is red and the mammal 2 is green. And when they come together, you always get a small part of CRTC1 and a slightly larger part, but it's always exactly the same genetic change. Could you go to the next slide, please? So one of the problems is, as Rob pointed out, is that this is a rare cancer. So getting enough patients or patient samples into any kind of study is quite difficult to do. And so there's a big range of in the studies as to how often this fusion gene is seen within salivary gland mucoepidermoid cancers. And within those studies, there is disagreement about whether it shows better survival rate or not. Next slide. At the minute, there is a gold standard test which is used by a number of pathology departments. And this uses a technique called FISH, which stands for fluorescent in situ hybridization. The downside to fluorescent in situ hybridization is that in order to pick up the signal, you need to be able to see chromosomes in the cells, because that's what it's picking up. And not all of our cells at any one time will be going through cell division, which is the point where you would see the chromosomes. You wouldn't normally be able to visualize them well enough. And also, as you can see from those little images there on the slide, it's showing you nice, bright red, green, yellow colors, and the blue background is sort of other cell nuclei. But it isn't telling you which cell. You can't, a pathologist couldn't tell from that which cell type is actually expressing the gene. So you get less information from it. You're limited by the fact that you need the cells to be going through division, and you get less information. PCR, which everybody knows about PCR since COVID, you can also use that to pick up the fusion gene and see whether it's there. But you don't get that spatial information. You don't get information about where it's expressed, which cells it's expressed in. Next slide, please. So other people have used classic immunohistochemistry, and this was in a paper that was published a number of years ago. This is suggesting that practically all of the cells, so the brown staining is the positive reaction, and this is suggesting that practically all of the cells are expressing the fusion gene. I'm going to put it out there that I think this is unrealistic. It certainly doesn't align with fish results that we've had before, that almost all cells are expressing the gene. So there are limitations to that as well. Next slide, please. So we actually carried out a systematic review of what was happening within the CRTC1 mammal translocation, just to see what was out there. So next slide. So most of them are, if you go through all of the literature, which is what we did, we found that most of the tumors were in major salivary glands, not surprisingly, and most of them were low grade. And that has an implication for whether you're giving survival rates. It's a good marker of survival rates. If all of your tumors are of one grade, then you're skewing the results somewhat. And they were carried out mostly with PCR or fish studies. Next slide, please. And as I just said, most of the studies came from favorable outcomes, but they were from patients, low numbers of patients, and mostly low-grade tumors. So you're kind of skewing the results rather than getting a good overall picture. Next slide, please. So this leads to sort of a very broad range of 5-year survival. And I think we can do better for the patients. And that was really what we were trying to do with the next study that I did. So if we go to the next slide, please. We tried to develop a new method of in situ detection. And as I said, it's always the same breakpoint. So there's exon 1 in red next to exons 2 to 5 of that gene, of those genes that come together. So we could design something that would only appear, see where it says 20 base pair exon to exon junction. You would only pick that up where those two pieces of gene have come together. You would only detect it there. So next slide. As Rob also said, it's very difficult to get hold of patient samples. And I'm really lucky. I've done a lot of work with people in Brazil. And so I can get hold of extra samples from there. So we had 30 cases to look at. Next slide, please. And then, so this is just showing the controls. The company that make the probe for us make us test to make sure the tissue is it will work well with the technique, that you won't get any false positive or false negative results because the tissue is degraded or whatever by the time it gets to. And this is fixed tissue, formalin fixed tissue, which does have implications when you're doing genetics studies on them. So if you see a little red dot, anywhere you see a little red dot, that's telling you that the gene is being expressed. And then the next slide, again, this is the start of looking at clinical cases. And you have to look quite carefully, but once you've got your eye in, you can see little red dots. And this is also more quantitative than the fish because you can count those red dots and then you know exactly how many times the transcript has been expressed. Next slide, please. So, and we found that all cell types that you find within a mucoepidermoid expressed the fusion gene. So that was new. That's new information that you can't tell from fish, as I said. Next slide, please. And it hasn't previously been reported in clear cells, which I think Rob mentioned as a cell type previously. Next slide, please. And we detected it in all grades of tumor. So these are low grade, and again, I put them at different. The pink staining is a normal H&E slide, which is what the pathologist normally looks at. And then there's the base scope assay, which is our in situ assay, and then a higher power to show that tiny little red dot in them. You can see very clearly in one of those cells. Next slide, please. And it's also in the intermediate grade, and the highlighted area has been, we've zoomed in on it so that we can see, again, the little red dots that tell us that the fusion gene is being expressed there. Next slide, please. And then, so this essentially was the sum of what we found. We found it in all tumors, low grade, intermediate grade, and high grade tumors. So from this study, we concluded that we are able to detect the fusion transcript, and we found it in about 30% of the cases. So could this be used as an alternative diagnostic technique? It's relatively straightforward to use. You don't have to have a specialist fluorescent microscope, which you have to have for the fish. It could be done in a routine lab by normal histology technicians. So it doesn't require the really specialist techniques that the fish does, and it gives you more information. We detected it in all grades, interestingly as well, which I haven't sort of said before, was we found it in direct contact with the stroma, so that's the sort of center of the tumor, and also because these tumors do have a little bit of a tendency for perineural invasion, so sort of to invade nerves, we actually were able to detect that in one of our cases, that the transcript was present where there was nerve invasion by the tumor. So it's giving us the pathologist much more information. Obviously, we need to do more studies, but I think it's a good step forward. So this study was a study that we did looking at the CREB pathway is one of the pathways that's been suggested that might be activated during the carcinogenic process of developing a mucoepidermoid carcinoma. The next slide, I think, is just the abstract of what we did. We took some 3-cell lines that were mucoepidermoid cells and looked at whether they were able to, so normal cells, it's actually quite difficult to get them to grow as little clumps of cells. But cancer cells are quite happy to go as little clumps of cells. So you can kind of tell the difference between the two. So the purple staining is just picking up the cells growing on a plate. And you can see if you add this inhibitor in, then you're preventing those clumps from growing in the cell lines that are expressing certainly the HMC2 expresses the CRTC1 fusion gene, as does the 292, but that's a lung mucoepidermoid. That's derived from a lung mucoepidermoid. So we were kind of comparing salivary gland with lung mucoepidermoids. Okay, next slide, please. And as I said, again, you can see the difference between the control. So the control doesn't have this inhibitor. It's not preventing that pathway from being activated that you would normally see activated in a mucoepidermoid cancer. And you can see the little clumps of cells are much smaller when you've got the inhibitor present. So really what we were looking at, we were trying to find was, is this a potential pathway that we could target for a new therapy? That was why we do the study. And these are normal studies that you would do. Sorry, go on to the next slide. It's fine. So where we've got pink staining there, what we're looking at here is migration of cells. So Terry told us that he has metastatic disease. And in order to have metastases, his cells from his original tumor have to start moving around his body. And normal cells obviously don't do that. And so one thing that we're looking at is, can we prevent that from happening? And you can see that there's less pink staining in the right-hand panel there than in the left hand, where there's no inhibitor. And so the inhibitor has prevented, to a certain extent, the migration of those cells. Next slide, please. Another measure of how easy it is for the cells to move within our experimental techniques, and a measure of whether cells will be mobile around someone's body, is to do a wound assay. So we essentially have a plate which is completely covered in cells, and then we make a scratch down the middle. And then we use time-lapse microscopy to watch the cells closing that gap. And obviously, if the inhibitor is there, we want it to take longer to close the gap than if the inhibitor is not present. And you can see over the different time period that has happened. Next slide. And again, this is looking at what we're really interested in. Is there are some cells within tumors called stem cells, and they're the ones that, if you have a relapsing disease, it's potentially because there are some latent stem cells that are just hiding within you, that the majority of your tumor has been removed and treated in whatever way that you've had your treatment, and then suddenly the stem cells decide to regrow. So we were having a look to see if the inhibitor had an effect on those two. And I think the next one is again a similar, very, very similar study, but looking at a slightly different pathway. This is looking at what we know as growth factors, which will help these cells to grow. So can we target the pathway that allows the release of this growth factor? It would normally bind to this TRKB receptor and then have its effect on the cells and allow the cells to grow. And you can see Vivian is on that panel too. So the growth factor is BDNF, and obviously the receptor it's going to bind to. And that's looking at the top two are looking at normal tissues. And you can instantly see that there's slightly less brown staining, a lot less brown staining in the top 4 little pictures compared to the bottom 6 pictures, which are mucoepidermoid tumors. So they're low, intermediate, and high grade. So you can instantly see that there's more of this particular growth factor there and more of the receptor, which will allow it to have its effect. Next slide, please. And we looked more closely using an electron microscope to see what was happening. So the same cell lines that I said before, the HMC2, which is a fusion gene expressing cell, 253 is another salivary gland mucoepidermoid carcinoma, and 292 is this pulmonary carcinoma, mucoepidermoid carcinoma. And cisplatin, most people will have heard of cisplatin. It's a standard chemotherapy drug, and ANA-12 was the new potential chemotherapeutic drug that we were hoping might have an effect. And I think, so the top 3 panels are just showing you what normal cells look like. And you can see that the cell is disrupted, completely disrupted by cisplatin. It's a dreadful killer of cells. And the ANA-12 did have an effect, but not as detrimental effect on the cell structure as the cisplatin did. Next slide, please. What did we do in this? Oh, this is a similar kind of wound closure, where we did the scratch assay, and you can see the difference between the control, which is the blue line, and the ANA-12. So there wasn't much difference there, but cisplatin had a real detrimental effect on the cells moving in both of the different cell types that we tried it there. Next slide, please. And this is again a similar technique using the migration assay. So you're looking for pink staining. There's a lot in the control. There's slightly less in the ANA-12. There's almost nothing in the cisplatin and almost nothing in the combined. So the cisplatin is much more effective than ANA-12, but it's really deadly, and obviously it has severe side effects for patients taking them, using that drug. Next slide, please. And these are pictures, as I've described for the previous study, looking at sort of growth of colonies. which is a growth of whether a cancer cell is really cancerous if it can grow as little colonies in the lab. And again, the cisplatin was much more effective at preventing colony growth than the ANA12. Next slide. And so these are little tumor spheroids that we can grow. There's absolutely nothing practically growing in the cisplatin treated spheroids in comparison to the ANA12. And interestingly, the combined, if you combine the two, then certainly for two of the cell lines, they were able to negate almost the cisplatin effect, which is quite interesting and requires a little bit more studying, I think.